Welcome everybody. This is the first video of the plant pathology training for Green School. My name is Mario Ortiz Castro and I'm going to be your instructor for this series of videos. My goal today is to provide you with concepts related to plant pathology and with some applied examples that you can identify some of the common diseases here in Colorado. So by the end of these videos or these presentations, you can recognize the type of issue that you might be having and apply some integrated management strategies to manage that issue. It is not my intention to describe all diseases in Colorado, as we might have a lot and it will depend on the type of plant and the type of garden that you have. But we actually want to provide enough information so that you are capable of identifying the type of problem, either in your own garden or when you are answering questions as a master gardener in the help desk. The first thing that I'm going to start with is with an integrated pest management statement. Pest management. So what is the approach of Colorado State University for integrated pest management? And as you might be reading, and you might have already observed this, but we at CSU employ a combination of cultural, mechanical, biological, and chemical practices. So we kind of approach this as a way of a combination of the practices that can help manage diseases and using the best available science without any bias. So that's important to consider, since some of the topics that we're going to cover today either apply the use of chemicals or the use of mechanical practices. So knowing that from the beginning will help us understand why is that we use this approach rather than a different one into management diseases. And we're going to start with this overview of what is plant pathology. And as you might already know, plants get sick too. So any part of the plant, as I'm showing you here in this sad corn plant, can get sick. So it can have either tumors or viral diseases, or on the leaves, even on the fruit, for example, or nutrient deficiency. Those are all things that the plant pathologists will help. Like, we want to have the plants healthy, same as you, so that's why we kind of study plant pathology. And plant pathology is the study of diseases and disorders of plants. So we want to make that connection, is that a disease is different than a disorder, but plant pathology studies both of them. So a disease is a harmful deviation from normal growth that is caused by an infectious agent. So we call an infectious agent something that is alive, and we call it biotic factors. So a disease is a detrimental relationship between a plant and something else that is alive, that is a biotic factor, and that will cause kind of it will be harmful for the plants. And we have three examples here of plant diseases. We have first Mersonina leaf spot, which is the name of the disease. And these are some of the symptoms that it causes here on the top left side of the screen. Mersonina leaf spot is caused by a fungi called Mersonina fungi. It is common on aspen trees. We also have here in the second part of the left side of the screen, we have early blight. And early blight is a common disease on tomato or potatoes and some solanaceous plants, and it's caused by a fungi called Alternaria solanae. And these are some of the symptoms that it will cause. Then we also have a third example here in the bottom part, that is fire blight. And fire blight is caused by a bacteria pathogen called Erwinia amylovora. So all these three diseases are caused by something else, some other organism that is alive. That is different from the disorders. Disorders are harmful deviations, so they are also harmful to the plants, but they are caused by non-infections, so things that are not alive. We call those abiotic factors. For example, we have end blossom rot that you might see. This is very common here in tomatoes if you're growing tomatoes. And the end blossom rot is related to a calcium deficiency and an uneven moisture in the soil. So this is not alive. This is an environmental factor or a nutrient deficiency factor. Same goes as the second example over here as iron chlorosis. And iron chlorosis is the deficiency of iron. So it's not so much that the iron is not available in the soil, it's that the plant cannot uptake it. So why can the plant not do it? Well, that's what we want to figure out. The third example that I brought of plant disorders is the sun scald. And this is very common in Colorado because we have, the sunshine state. We have more than 300 days of direct sunlight. So that will have some effects on the plants. And in this case, on this paper, it's burned on this side. This is safe to eat since this is not like it's an abiotic factor. So you will just, if you wanted just to eat it, you will just cut this part and eat it up. So we'll talk later about those effects, but the idea is that plant pathology will study both diseases and as well as disorders. So why do we study plant pathogens? Why are we here and why are you listening to this video? And I will give you some examples of why are we studying plant pathogens, but and this is a good reference, this is from the movie Moana, the Disney movie Moana. If you have kids or if you have grandkids, or even if you are just like me that you like Disney movies, you will see that in this movie, the main plot is that they live in an island and the coconuts are having a disease, and the livelihood of the entire village depends on what they use for the coconut. So in this case, I'm just giving you an example, and although this is not real, the coconuts are suffering from a disease. In this case, it's a vascular disease that went all the way from the roots to the actual coconuts, and they are turning black. So you can see that the study of plant pathogens have always kind of been impacting how they impact the humanity in that sense. But we study plant diseases worldwide because they have a real impact in the world. Losses from plant diseases can have a significant economic impact. So if you think about money-wise, and this is because they will cause either the reduction of the income for producers, so for people that are growing them and that they are selling their products, but as well for distributors, because they will have to implement other source or buy some other chemicals or buy some other equipment to manage the diseases. But then it will also influence the consumers because the prices will go higher. So this is in a large scale, let's say a large scale production of some sort of crop that you eat. If it's impacted by a plant disease, then it will be impacted in the high prices at the end. If you're thinking at a smaller scale, for example, you go to a garden and you want to buy a plant, but the plant is sick, and you still decide to buy it, then you of course will need to first nurture that plant because that the plants become healthy, and that will of course kind of like relate that you will need to buy either a chemical or a different type of fertilizer or a different type of methods for environmental. So that in itself is a current impact of plant diseases, because you will need to take an extra step to take care of the plant, rather if you just start with a healthy plant from the beginning. It is also important to consider that the estimated overall losses of plant diseases is between 10 to 20% of yield loss. So if you, in any given year, you will get, let's say, this amount of produce, or let's say in the case of gardeners or flowers or vegetables, just by having a disease, it will have a 10 to 20% of yield lost. This will also include post-harvest diseases, as well as diseases that the cumulative damage might be large, and cosmetic issues for ornamentals. These losses are not accounting for the losses, for example, in developing countries that they don't have resistant varieties, or they don't have chemical control available, or they can also not afford them. So the yield losses could go all the way higher, from 50 to 60% in countries that don't have the right science to be able to manage those diseases. So if you are not convinced yet that the plant diseases are important and that why are we studying it, I'm going to give you an example of how plant pathogens have impacted the humanity. And the reality is that plant diseases have always influenced the course of human development, because as you might know, we have used plants since the beginning of times. This is a masterpiece, and it's called The Potato Eaters. And it's making a reference to the peasants, the Irish peasants that were eating potatoes. And it's a famous painting by Vincent van Gogh. He dated in April of 1885. So why I'm bringing this picture today is because I'm going to show you one example of how plant pathology and plant pathogens have changed the course of humanity. And what happened, and this is making reference, as I said before, of the Irish eating potatoes. What happened is that there was an economic boom in England thanks to the island potatoes. So in the early 1800s, the Irish population was similar to what we see in many poor countries today. But at that time, England controlled Ireland. So they then introduced the potato from the New World. So from South America, they introduced the potato into Ireland, and voila, farmers could grow 3 times as much food per hectare because all the conditions that were in Ireland were favorable to grow those potatoes. And the population in Ireland doubled in 30 years. So it not just grow in terms of the crop of the potatoes, but also in terms of people. So this was considered a period of economic boom for the English who controlled Ireland. So not for the Irish itself, but for the English that had all the control over the Ireland. But then in 1844, you have the Irish potato famine. And the disease was called Blight of potato, and it appears in the US in 1844. And in 1845, an epidemic sweeps Ireland, infecting nearly every field. So every single field was infected with this disease. So this not only caused the livelihood of people that live in there and that work the land, but many people starve and die because, well, they didn't have means to eat, but also they were evicted from their homes and they couldn't pay rent. So they just live in the streets, and it was just definitely not a good situation for the Irish. The worst part of that is that the British response with emergency food was very slow because they didn't want to damage or disrupt the economic opportunities that they were having. So even though they were not having the problem of the Irish potato famine, in England, they were still charging rent. So people didn't have anything to eat and didn't have anywhere to live. So the British response was slow. And also British didn't like Irish. So that was also something else that influenced the development of this disease. So the response was slow. But then this Irish potato famine had a legacy even on social and on cultural cues, because more than 1.5 million of people died from starvation and almost 2 million immigrated from the United States to the United States. So immigrants really changed the demography of the United States, and you probably know or have family or know somebody that came from Ireland due to the Irish potato famine. This, of course, changed the political relationship between Ireland and England, resulting in the Ireland independence in 1918. So it's kind of just one pathogen, just one disease caused all these disruptions, social, political, cultural disruptions, and even changed the demographic of an entire country. So what was the causal agent of this Blight of potatoes? Well, the causal agent is a pathogen that is called Phytophthora infestans, and it's still a problem today. So it's not something that it has been eradicated, even though it was in the 1800s, it's still a problem today. And if you want to learn more of how Phytophthora infestans is managed and control and everything about Phytophthora infestans here in the United States, he have his own website, usablight.org, if you want to go there and kind of like learn more about it. But the thing is that Phytophthora infestans is a pathogen, and it requires, or it likes very wet environments. So that's why Ireland, that rains a lot, was the perfect kind of a storm to develop this disease. So what factors influenced this disease? Why was there such a big disease and such a big impact, more than that it was wiping the entire field? So that's what we're going to talk about. The first factor that influences disease was the pathogen. So we have an organism that is what we call a water mold or an oomycete. It's not a fungal pathogen, it is an oomycete that reproduce very fast by what we call zoospores. So those zoospores reproduce very fast in the water. And they are either air or water dispersed. So within a week, you could have millions and trillions of zoospores that could be infecting every single field. Then we have a host, and in the case of potatoes are vegetatively propagated. That means that the host doesn't have any genetic diversity. So one potato plant is exactly the same genetically than the next potato plant. So that kind of like increase the probability that a pathogen will wipe out every single plant. And then we have an environment. And as I mentioned before, Ireland has very wet environment. It rains a lot, but it also didn't help the cultural or farming practices of the time. So that also kind of helped to develop this disease. And contrary to what the name says, potato leaf blight, this disease can affect any time during the season. So not later at the season, but any time during the season. So this is not something that, oh no, it's gonna get, let's say, if the season starts in May, this is just as an example, oh no, it's gonna get in August or September. No, it can infect any time during the season. So those three factors, the pathogen, the host, and the environment, really influences the disease. And this is the first concept that we're gonna talk today in plant pathology. This is what we call the disease triangle. And pathogen, host, and environment, when they align together, when they are present at any given time, then the right conditions will form diseases. And as I mentioned before, once they intersect, they will have disease. But if, for example, you have a host and a pathogen, but you don't have the right environment, then you will not have disease. You will need to have the three components. These are absolutely necessary to have. Some people and some researchers also refer to this disease triangle as the disease pyramid because they inserted the time factor as another component of the triangle. But the thing is that at any given time, when those three, the pathogen, the host, and the environment are together, then you are gonna have disease. So in the presentation and in this video, I'm gonna use either disease triangle or disease pyramid interchangeably, but they refer to this principle of disease development in that case. So now we have talked about why do we study plant pathology, right? Why do we study plant pathogens and their importance? But then we want to say now, why is it so important to accurately diagnose plant problems? So why is that we're here and understanding what plant problem it has? When we want to figure out what's wrong with my plant, so we want to figure out what is actually happening to it. So I'm going to say, and I'm going to first say if these symptoms are representing the same disease. At first hand, and if you just see these pictures, you might be like, oh, yes, maybe it is representing the same disease or maybe not. But the reality, and this is going to be kind of like showing you that these two represent two different diseases. These two pictures represent one disease that is called gray leaf spot, and it's caused by a fungi. Cercospora siamatis is the fungi. And these two other pictures represent bacterial leaf streak that is caused by Xanthomonas vesicula Pathology vascularum. And this affects corn. This is what later in the season when this bacteria is on, how it can affect it. And you can see that the yield is going to be reduced because the flowers cannot go into the tassel, and therefore you cannot have a good production of the corn. This is how the bacteria looks on the back and on the leaves, and that I'm showing you. So why I'm saying all this is because, and this is a little bit of reference, this bacterial leaf streak is an emerging disease on corn. And it was first reported in 2016 in Nebraska and then in Colorado. And we were probably the first two states that kind of like say, oh, we have a new disease. The problem was not so much that they identified the disease, is that growers were thinking that it was gray leaf spot and they were applying fungicides because it's a fungal pathogen. But they were just losing their money because it's a bacterial pathogen. So bacteria and fungi are different. So the methods that you're going to apply to control or to manage fungal disease are very different than the ones that you use for a bacterial pathogen. So that's why it's so important to accurately diagnose the plant problems. So if we know the identity of the causal agent, we can better understand his biology and offer effective management strategies. So although the growers were applying a management strategy, it was not effective. It was doing nothing to the bacterial leaf streak. So that's why it's so important to understand and to diagnose those plant problems. And I know that in another module, you talk about plant diagnostics. So we're going to cover this topic as well here. A second reason why it's so important to diagnose plant problems is because we need to do an early detection of exotic and or devastating pests and pathogens. So we don't know the risk that a new pathogen poses to a new area. So we need to have quick actions to prevent that further spread into many plant species. And showing you here an example of Dutch elm disease. And Dutch elm disease is a disease that has killed millions of American elms in the urban landscape in the last 50 years. So this is how the tree looks. It's just devastated. And this is how the fungi looks under the microscope on a petri dish. The problem is that because these plants have not been exposed to the new pathogen or the pest, then there is often no genetic resistance. And so there is no natural predators to those exotic pathogens. So it's not just that we need to do the early detection, but also there will not be a natural enemy for that pathogen in that case. So that's also exacerbating that problem. So that's why it's so important to also understand why do we want to diagnose those plant problems. So how can we diagnose plant health problems? Well, we use the plant diagnostic process. And this is a process that is used for the identification of plant health problems. So we're going to use several tools. We're going to use plant sciences, specifically plant pathology and entomology. And you already have a module on entomology, so we're not going to cover that today. But we're also going to use detective skills. So it's going to be kind of like CSI for plants, so because we want to figure out what's wrong with my plant. And for this, the remainder of the video, we're going to talk about this plant diagnostic process, and I'm going to show you a systematic way of how you can do it. So the plant diagnostic process is sometimes it can be daunting and overwhelming because there are many different problems that can affect the plants. But it's also hard because plants can have several symptoms, and those symptoms can either mimic biotic or abiotic problems. So things that are alive and things that are not alive. And the issue is that things that are not alive, so abiotic factors, can mimic symptoms of things that are alive. So it's kind of hard to diagnose. So that's why practicing is going to be important and understanding what's going with the plant. The first step into the plant diagnostic process is to identify the plant. So we want to know what's our baseline. We want to know what we're working with. In identifying the plant as a first step, we need to know the difference between common name versus scientific name. So the common name is the name that usually people will talk about. Oh, we have cyniat, or we have several flowers. So this is something that the common general public will talk about. Right? The scientific name is the name that is given the two-name Latin binomial that the scientific community use. And that's why it's so important to know it, or at least research it, so that you know what your baseline is. And this is because sometimes in different part of the country, even in different part of the world, one single plant can have many different common names. And this is an example, the Douglas fir, Oregon pine, and red fir. They have already 3 different names, but it's referring to the same tree, which is Pseudotsuga messiensis. This is kind of the picture of the tree. So if you see this tree and somebody tells you, oh no, I have a Douglas fir, and somebody else told you I have an Oregon pine, and you might think they are talking about a different plant, but it's the same plant. So you can have a baseline, as I mentioned before, of what you're working with. In identifying the plant, it's also important, if you can, to know the variety of the cultivar. And that's because sometimes variety of cultivars are developed to have different features than the original plant. This is an example of an old gold juniper. And if I just see this plant, this juniper that is yellow, without knowing that this is an actual variety, I will think, oh no, this plant has some sort of problem. It doesn't have enough nitrogen, or it has iron chlorosis. I'm just going to start making assumptions based on what it is, but I will not know that the plant actually looks like that. So it's something that we need to consider when identifying the plant. Whether sometimes if you go to the garden centers, they will tell you the variety or the cultivar, so that's important to know. Then in the second step, so we know already what plant we're working with, and hopefully we have already researched the scientific name, so we have a baseline. Then we need to identify what a healthy plant looks like and what's normal versus abnormal. And this is because sometimes, and you will see this often, that you have a healthy plant and it looks bright, looks green, it doesn't have any symptoms, but then 10 days later, the plant looks like they have these necrotic spots and it's unhealthy. So you definitely know that something happened here to your plant. From healthy to unhealthy, this is something that is affecting our plant. But that is different than normal versus abnormal, because sometimes you're going to see that you are going to look, the plant doesn't look healthy, so it's unhealthy, but it's completely normal. And this is, for example, when in conifers, the needles in the fall are losing the needles. And some people will call us and say, oh no, my evergreen is losing the needles, what do I do? And this is the fault, and that's a completely normal process, although it looks unhealthy. So that's important to know. And then, of course, the abnormalities are gonna be the ones that caught your eye. Like these scale insects here on this conifer or on this pine is gonna be very abnormal and actually gross. So you know, and you're gonna be able to know because you always check your plants. In that case, you always scout for those plants. So identify what healthy versus unhealthy and what normal versus abnormal is gonna be crucial as well. So that's why it's the step number 2. So now we're gonna move into the step number 3, and this is where we're gonna spend the majority of our time in these presentations, is to identify the problem. So what is actually what is happening to my plant? And hopefully by the end of all these modules, you will know what type of problems you can have so that it's not just like, oh, I'm going blind with the problem that I might be having. So in identifying the problem, we're gonna have the difference between symptoms and signs. So what is a symptom, what is a sign? And then we're gonna also cover the difference between biotic and abiotic problems. So we're gonna cover the difference between organisms that are alive and things that are not alive that can cause a problem on the plant. In these 2 pictures I'm showing you here on the bottom part, these are some spores of Alternaria solanae, and in this one particularly in the middle, it has just germinated and you see the hyphae. So we're gonna talk about what is it that you can look for between symptoms and signs first. So we're going to start with the symptoms, and symptoms is a plant reaction to the activities of the pathogen. So it's a plant, the plant is actually doing something, is showing you something due to the activities of an external factor. Symptoms, as I said, are changes in the plant's growth or appearance in response to the causal factors. And these symptoms can either be localized, for example, these spots here on the leaf or these galls that are usually on the base or on the crown root. These are something that is just in that part of the plant and it doesn't move farther, for example. But you could also have systemic symptoms that affect the entire plant. For example, wheels, like I'm showing you here in this picture, of mosaic patterns that are everywhere around the plant. The good thing about symptoms is that it's something that you can see. So the plant is telling you something is wrong with me, please help me. So they are very important in the diagnosis. But the bad thing about the bad thing about symptoms is that different pathogens and abiotic stress might cause the same symptoms. So whether it's a fungal or a bacteria or even a nutrient deficiency may have the same symptom. So that's when it's problematic to just rely on symptoms to do the diagnosis. And usually symptoms progress, like they start on one leaf and they move into the next one because the plant is trying to ask for help. So this is the plant reacting to whatever is happening to it. These are some examples of symptoms. We have cankers, we could have galls, soft rots, chlorosis, blights, mosaic patterns, or leaf spot. And we're going to talk about some of them in here and define them so that when you are looking at your plant and you want to define what your symptom is, you know the actual terminology. So the first one is the chlorosis, and chlorosis is basically the loss of the green color. We know that the plants have chlorophyll, and that's what it gives the green color to the plant. So when the plants start losing that component, that chlorophyll, they will either start turning yellow or only along the veins, like I'm showing you here in this picture. But chlorosis is losing the green color in the plant. So why are you losing the green color? We don't know, but that's what the plant is telling you. Something is wrong and I'm losing my green color. That's not normal. The second concept we have is the mosaic, and these are variant patterns of light and dark plant tissue. So you can see here in this picture, so you have light green, dark green, same as on these leaves as well. Usually mosaic patterns are related to viruses and they are systemic. So it will be in the entire plant. So that's why you will start to see it in all the leaf in that case. We could also have leaf spots, and these are limited, discolored disease areas of the leaf. So the areas, the leaf itself, I'm showing you kind of like, this is a dead spot, this is a dead spot, and they are very, very kind of characteristic. So you can determine that this is a leaf spot, and this leaf spot, something is happening. These are very, this is probably one of the most common symptoms, those leaf spots. And those spots are usually necrotic. And necrotic means that it's a death of the plant tissue. So you can see in this right-hand side of the screen that this is part of the leaf that has died. So this is a necrotic leaf spot. We could also see on this other plant in the middle that the necrosis could just be in the entire leaf or only on the edges, as I'm showing you in the left side of the screen over here. So leaf spots could usually be necrotic as well. Then we have blight, and blight is the sudden rapid death of tissue. And it's usually not like spots, like very punctual, but it's actually around the end, for example, of the tip of the branch that is actually sudden, just died. And you can see it on woody plants here that the tissue just died, but the leaf stay on. So this is also something important that is so suddenly that it just happened. We could also have wilting, and this is the damage of tissue that causes the leaf to become flaccid. So they are like lumpy, and this is the result of a water loss or the restriction of water movement. So the plants are droopy, and this is usually a symptom sometimes that when you haven't watered your plants, they are gonna look like that. But some pathogens can also, even if you are watering your plant, can also cause the wilting of the plant. Some, especially soil-borne pathogens, will cause those wilting symptoms. But again, this is always on the plant. So the plant is telling you that something is wrong. We could also have some canker, and canker is the sunken discolored areas that are on the stem. This is often associated with the death of the bark or the cambium in woody tissue. So you can see these three different stages of the canker. This is usually when it's starting at a later stage, and this is when the canker is like very bad in that tree. We don't know what the cause of the canker is, but we know that the canker is a symptom of something that is wrong with the plant. We could also have stunting, and this is the lack of the plant growth. So compare these corn leaves in the back to these ones over here. They are significantly smaller than what they are supposed to do. They also have other problems and other symptoms like that, yellowing, striping, but the stunting is what we are referring here. You could also have galls, and these are localized swellings. So what happens is that you could have an insect or a pathogen that hijacks the hormonal system in the plant, and then this is a hormone imbalance, so the plant starts either swelling, in this case, and creating those galls. So because it's a hormonal imbalance, it's kind of like having a cancer in your own plant. Could also have distortion, so malformed plant tissue that this is not supposed to look like that. It's not even have any leaf, it's not creating any leaf, or they are curling up. This is called a distortion. We could also have some witch's brooms, which is the proliferation of shoots from a growing point. And this is malformed plant tissue. This is an example of those distortion. We could have those witch's broom on fir or spruce or an evergreens. So they come low, kind of just grow there, but it's not the normal within the evergreens. Or you could also have it on deciduous trees. For example, the hackberry could have those witch's brooms as well. What is causing the witch's brooms? Again, we don't know, but the plant is telling you something is wrong. Please help me. Symptoms can be found anywhere within the plant. They can be found on the leaves, on the stems, on the roots, flowers, fruit. So it can affect any part plant, and that's when you should start considering. So what part of the plants I am seeing? I start seeing it on the fruit, but I could also see it on the leaf or on the stems, and even on the roots if you pull them out. Please be aware that the symptom varies, and there is some what we call symptom progression. So it could start, for example, on the leaf and then move vascularly, or it could start on the roots and move outward. So you need to consider that when you are looking. And please be also aware that environmental conditions, host genetics, and the physiology can affect the appearance of symptoms. So you could have the same type of symptom in two different plants looking very different because of the host genetic or the environmental conditions. And plants can have more than one problem at the same time, showing you just one symptom, but it can have different problems. You could also need to identify primary from secondary symptoms. For example, the primary symptom could be that the plant is not, if you see this plant in this case is wilted, the primary symptom could be that the plant is not absorbing enough water. But what you are looking is at a canker. So the initial symptom is that the plant is wilted, it cannot acquire enough water, but the secondary symptom is the canker that creates for kind of like dying of that bark tissue in that case. So this is something that also needs to be considered. So we're now going to talk about signs. So we talk about symptoms. Those are the plant reactions to the activities of the pathogen. But the signs is the actual organism that is causing the problem. So this is the actual living thing, in this case, that is causing the problem. Signs are either microscopic or microscopic. They are the product or they are a part of the structure of the pathogen or the pest. So what I like to call them is like they are the fingerprints because it's the evidence of the pathogen. Therefore, because you can also see the evidence of the pathogen, they are a much better diagnostic feature than the symptoms because it's not just the plant having it, actually the pathogen. But of course, only biotic agents can have signs. So something that is alive is something that is going to leave you either a product or the evidence of it. We have examples here of signs. We could have mycelia, like on this grass. This kind of look like a web on a spider, but no, it's actually mycelia that is growing on the grass. It's wet over here. We could also have bacterial ooze. This is sometimes hard to see, but if you catch it at the right time, you will see kind of like from the plant coming out a gooey part, and you can see it here under the microscope over here. You could also have spores, in this case, the conidia or fruiting bodies or nematodes that you could only see under the microscope, or spores. The spores are usually very easy to see because they are fluffy in appearance and dry. But these are signs. So this is the actual causal agent. And we're going to go a little bit more in depth of the type of signs that you can find. So you could have the ooze, as I mentioned before, it's kind of like a slimy light droplets, and they are caused by bacteria. These are 3 pictures showing you 3 different types of ooze. So you could have it on the bark, on the wood over here, or in this other picture, you could even have it on the fruit. And I wouldn't recommend eating this fruit with how much ooze it has, right? It's not good to have it in that case. We could also have some reproductive structures that we call fruiting bodies, and these are specifically for fungi. They could be microscopic, such as the mushrooms or the cones that we're looking at here. So the mushrooms in the kind of like in the lower part of a tree is a sign that the plant is the mushrooms are eating the woods, and it's what we call wood decay mushrooms. We could also have some of the macroscopic reproductive structure, such as this one here, that is type of rust in the hawthorn. This will be the macroscopic structure that you are going to see. You could also have fruiting structures that are specific to fungi, but that are microscopic. For example, the Cleistotaceae, you're going to see these on powdery mildews using even just a magnifying glass. You will be able to see it. Under the microscope, you will be the cervuli, bristles, or the Perithecea that have kind of like a pear shape that you can observe over here. But the idea is that these are the signs of something, the signs of the fungi in that case. You could also have some fruited structures that are embedded within the lesion, like on this canker, these dots over here are those fruiting structures. Or on this lesion over here, those dots over here are also the fruiting structures of the pathogen. We could have spores or spore masses. Under the microscope, you will be able to see these both, but if you don't have a microscope or a magnifying glass, you can see them with your naked eye, kind of like the fluffy appearance that I was mentioning, or like on this peach, those spore masses. I already showed you this picture before. This is the conidiophora growing from a fungi infection. It's kind of like a lollipop shape, and this is discolored germ disease. And I'm showing you here because there are many different type of signs that you can look for, right? If you have the right conditions and the right tools. We could also have mycelium, which is the thread light. This is the vegetative growth of the fungi. You could have it on the vegetable, like on this carrot, or on the grasses that are growing. And this is what you can actually see that this is a sign. So what's the difference between symptom versus sign? So you are gonna see the effect versus cause. Symptoms is the plant's response to stress. So this is what we call the effect, because the plant is showing you that something is wrong. But the sign is the reproductive or vegetative structure of the causal organism, which is the actual cause. For example, this Rhizoctonia here, this is the mycelium in the root. This is actual mycelium. So I'm looking at the pathogen itself. So if you want to know, or if you want to have like for a better difference between symptom and sign, let's say, and this is an analogy that I'm going to use. You have a fever and you are like feverish and you don't know what it is, but you have a fever. And usually the fever is the symptom, right? So you go to the doctor and the doctor tells you, oh, it's because you have a bacterial infection. And this bacteria is the one that you are having. Let's say E. coli, for example. Therefore, the symptom is the fever, but the sign is the actual bacteria. So same goes for the plants. The symptom is the effect of whatever the plant is showing you. In this case is wilting, like on this example, but the actual organism is the fungi Rhizoctonia solanae. So that's how we can usually identify between symptom and sign. We're gonna have this kind of like an activity. So this is just for you to practice. Is this a symptom or a sign? And this is a lilac. We already talked about this. Gonna give you 30 seconds so that you can think about whether is this a symptom or a sign. Okay, so this leaf, I hope you have time to think whether this is a symptom of a sign. This over here in this picture is a symptom and a sign because you see in the talc powder, right? But this talc powder like is the actual fungi, but it's also a symptom. And we're going to talk a little bit about this type of disease later on. But if you see on the back end of the leaf, you will also observe this Cleistothecia over here. And this is definitely the sign of a pathogen that is over there. So we're going to talk about those differences between symptoms and signs throughout all our videos, but this is a good activity to start. I have another one here. So we're going to start with this one. And this is a crown gall. And as I mentioned before, the galls are symptoms. So this is tissue from the plant. So this is not the pathogen. The pathogen is making the plant to create a gall, but this is plant tissue. So this is a symptom. This is a mushroom, and mushrooms are what? Signs, right? So signs, because this is the actual fungi. And this we have this last picture of necrotics. So this is a necrosis or necrotic leaf spot, which is also a symptom. You are not looking at any pathogen in there. Now, if you look under a microscope, you might be able to see those fruiting bodies that I was saying that are embedded, but just with our naked eye, we're just looking at a symptom in that case. Please, if you have any questions, write them down and bring them to the live session that we're gonna have so that you can get your questions answered. Thank you. I'll see you in the next video.